But while I think we could start now, and I'd like to thank you all for contributing to this event. We'll start by actually Dimitra will give a few words about the housekeeping things, and then I will just introduce the program. We cannot hear you, Dimitra.
Yes. Okay, good. Yeah, hello everyone, and welcome to the Diverse Xenewatch online event. Today we will communicate our solutions for genetic monitoring of forest trees in the Baltic Sea region. Yeah, my name is Dimitra. I'm a member of the Copenhagen University team. And before I give the word to Eric for a more detailed introduction, I just wanted to give you some practical information for the meeting today. Yeah, as Jill mentioned, parts of today's presentations and discussion will be recorded. Also, presentations today are either 15 or 20 minutes, including questions. My colleague Maxi and I will try to write you a message on the Teams chat when you have 5 minutes left. And if all your time is up, we will kindly let you know. Yeah, please consider to leave some time for questions if possible. And if there is not enough time to answer all the questions, you are encouraged to write your question in the chat, and we'll gather all the questions, and we'll try to get back to them during the discussion. Yeah, I hope we have a really nice event today, and I think that was it for now from me. Eric, you, whenever you're ready.
Yeah, yeah, I'm ready. Can you see my screen now?
Yes.
Okay, so in correct mode and everything. Okay, so thank you for joining this. The plan is that we'll discuss in this event. We'll work on genetic monitoring of forest trees in the Baltic Sea region. Okay. Are my slides changing? Okay, it's good. The point of the party is, of course, that genetic diversity is important as building block for breeding and as essential for the evolutionary potential of species. We saw that very recently with the ash dieback, where the emergent infectious disease, Hymoscurus fraxineus, are damaging the ash forest. But here again, we see that our diversity in tolerance actually allows using diversity as building block for breeding, but also as an important part of the ability for the species to evolve. And then, of course, also the diversity are recognized with the conservation of biological diversity as an important part of biodiversity, and therefore it's something that we should protect. And the question is, how do we do that? How do we monitor? And as we would say, so the objective of today's discussion would, in a nutshell, would be how can we, in a feasible way, monitor the level and dynamics of genetic resources of our trees, and in a way that we can infer on whether the resources are in good shape. So I took this from actually Solveig Pors Nielsen, who are from one of our previous meetings discussing what do we really need. So what we need to know is, does conservation effort works, or is there a need to do some actions in terms of protections or diversifications or modified management? Okay, so this would be hopefully what we discuss today. There's I think a really good input today. I'm really happy. We get input from one of Jesper Sorskud from the Danish Nature Agency, who will tell something about the Danish conservation programs. We got the input from Rosario Garcia Gil from SLU, that will tell us something about the experience from with studying genetic diversity in Sweden. And we have Barbara Fussi from the Bavarian Office of Forest Genetics that will tell us something about the experience from other parts of Europe. And then also very interesting, we have Birte Pakul that will share experience with the integration of genetic monitoring actually in national forest inventory. Then after this important input, there will Darius Danavicius from that is coordinator of this program, will tell something about a bit about more the details of the program. There'll be a short break then, and then Jill Olafsen from university here will tell something about our plans for how actually to implement this in a very particular way. And then hopefully after that, there will be some discussions where we can try to see what we can learn from these things. This would be the plan. So the point is dig diversity, and we see dig diversity as resources. And of course, that's diversity per se. But it's also a resource because it's something that we can use in some ways. So when it comes to defining our resource, it's about diversity, but it's also important diversity, so to speak. But when it comes to what we want to conserve, it's not only diversity, but it's actually also the patterns of diversity. So for monitoring, it's important to discuss how can we monitor how diversity is separated between species, between populations within species, and also, of course, between individuals within populations. So I suppose the point of departure for discussion is also how do we monitor diversity per se, but also how the diversity is distributed at these different levels, because the levels are important when it comes to how to manage diversity. Okay. Another thing which is I think is important in discussion is whether we should only conserve the dig diversity as can be measured by the DNA polymorphisms, which what we normally do in static conservation or preservation, where the focus is really to make sure that we don't lose things, we just keep things as they are. But the alternative approach for conservation is what is normally termed as evolutionary conservation or dynamic conservation, where the point is actually we're not only focused on the diversity per se, but also the processes that continuously shape these this diversity. So the idea would be that actually, since species are exposed to competition and new environments and also new pests and pathogens, the idea would be that we actually want gene frequencies to be allowed to change. And that means that we have an approach which you could call more evolutionary conservation. So we actually should ensure that the species maintain adapted to the ongoing environmental changes. And a focus in European conservation programs is actually on these evolution programs mainly, and I think for good reasons, because the changes are so rapid. I took this slide from the most recent IPCC report, which shows the colors here are indicating the temperature, and we have then a time scale, and the point would be how much change we can actually expect to observe within lifetime of a human, or actually what we have already observed. And taking into account the age of trees, of course, these are really major changes, which also all speak in favor of trying to think about how we do conserve things, but also in evolutionary perspective. So actually, this idea about evolution is kind of, you could say it's kind of complicated. How can we conserve the evolution processes? And I just took this slide, which just says that actually, when it comes to understanding the evolution process, actually it's not that more complicated than we could somehow actually put them into three processes. We have the natural selections that could lead to local adaptation, which would be important. Then we have the small population sizes, which is creating a lot of problems because of loss of diversity, inbreeding, and dig drift. And then we have the gene flow that somehow would counteract the loss of diversity and inbreeding developed in small populations, but on the other hand also would counteract local adaptation. So to say that that these processes are something that we can consider when we discuss how to monitor the programs. Again, here I tried to say that different kind of approaches for conservation, but today we'll mainly discuss on how we actually focus on this kind of evolutionary conservation, where we actually look at the diversity but also processes. And as I said already, the implemented programs in EU, and we heard about that, is probably, as I said, mainly the in situ. I took this slide from the Eufogene homepage about where we have the conservation stance as documented in EUFGIS. And on the left we have for Quercus robur, which is one of the species in this program, and on the right we have the Tila cordata. And here we have the conservation stance, which would be a key factor in discussing how to monitor conservation. But that's one option at least. And just to say these two examples, we could say that for the oak, there's challenges, for example, with the acute oak decline, which is a concern. But there's also processes related to how the species, the robo, would actually hybridize or integrate with Petrea, and how that could actually seen as a way for increased adaptation in the future. So these are processes, just to say that there are different processes here which are important to observe. And then another case with the Tilia, we have a situation where actually the warmer climate probably will influence the ability to reproduce. So there will be other processes that could be important. So what kind of toolbox we discuss a bit today? The toolbox we have, of course, we can go on and look whether the species is present, whether we have natural regeneration and how the phenotypes are. We could do that in conservation stands to show it, or we could do it in plantations, across landscapes, or in common gardens. And we can also look at the DNA variation in neutral marker or genes as social fitness. Today we will focus on actually the last part, discussing how to implement programs for studying the DNA variation. But another part is also looking at phenotypes is also an important part of actually the gene watch process, as we can discuss. So this was a very brief fly into just some of the idea about the conservation and the idea about monitoring here. I put some questions here that hopefully we can during this morning can address and discuss. So one thing I put up here: do we need to preserve both genetic diversity per se, or should we really focus also on the evolution processes? And if we do that, which process would be important, and how can we monitor them? I think also important discussion could be how we can really benefit from this idea that we work here in this project across countries. So cooperation between countries in a regional scale, but also in European scale. How can we benefit best from that, and when we make this genetic monitoring program? To what extent can the data actually be used to study the shape of the genetic resources? How do we ensure that the monitoring actually can feed into some decision-making process? And that could also lead into a discussion whether we should, pro and contra for actually monitoring only conservation stands versus monitoring across landscapes and so on. And this is things that will be addressed during this morning's presentation also. And then hopefully also there'll be time for we get some feedback to what we have proposed so far for joint program. So this would be my very brief introduction to this day, and we have the program here with that I look very forward to, and as already mentioned, Dimitra will try to keep time, and now probably she's giving me a signal, so I will pass on and stop sharing and give on and pass on the word to the first session where we will have examples, and the first presenter will be Gunnar Friis-Bosorski from the Nature Agency. So thank you, and please, Gunnar, I'll stop sharing.
Thank you very much.
Thank you, Eric, and great timing. Yes, Gunnar, whenever you're ready, you can share.
Right, to share a presentation.
Yeah, looks great.
Okay, thank you very much. My name is Gunnar Friis-Bosorski, and I'm working in Denmark as part of the Danish Nature Agency, and have been responsible for implementing the gene conservation strategy for more than 25 years. And as an extra historical thing, you should know that actually Erik Kjær was one of the persons who formulated the strategy back in 1994. So now we must see if Erik is satisfied with what has happened since then. I'll try to tell a little about what has been done. And the question today also is, how do we know if it works? And that's, I think, this project we are discussing today is actually very important to give us some hints about that, and also, of course, what to do forward. In 1994, there were some questions raised and some decisions made, and one of them was that it should be the evolutionary conservation that we would follow and we would think was most important. We covered 75 species, and that means that we covered the native forest trees, but also all the shrubs. And then we have a lot of exotic species that are very important for the forest production. At that time, knowledge was limited, but also we expected the knowledge to increase. So we kind of started out knowing that we had to do something, but perhaps not knowing exactly all the details that would be nice to know. So there was some kind of flexible thing that we thought that it would be wise to do what we did, but also be prepared that the strategy should be adjusted. It was pointed out that we could implement within 10 years, and well, we almost succeeded in that. But we also decided that to make a strategy that could secure things for more decades, then it had to be implemented where the ownership was state-owned forestry. And the focus was on in situ for the native species and ex situ for the native and introduced species. But the ex situ conservation in the implementation phase became more static in the sense that, well, I'll come back to that. And then I've shown you a map, which we called gene ecological zones in 1994. And actually, you could raise the question if they actually, how much genetic zones they actually are, because they also reflect climate and soil types and things like that, and reflects the distribution of species in Denmark. But that was somehow the way we looked up on our point of departure, and then we started implementing. Then the objectives we had was, as Eric also mentioned just now, something about this potential value, and also to secure a kind of buffer if the environment should be changing, and also as building blocks for breeding. And now 30 years later, you could say, well, it's still a very good question. What is the value of trees? What is the value of forests? In Denmark, there's been a change. You can say that production values, of course, has some importance, but also biodiversity or just having forests for its own sake or for recreational uses has become even more important than it perhaps was considered in the beginning of the 1990s. And now 30 years later, I must also say that conservation actually rarely contributes to breeding. When we are looking for plus trees are having breeding, we are not just limited to finding the trees in our gene conservation units. We are looking everywhere where we can find relevant material. And that's also some important point of view that perhaps we should look up on the gene conservation units as a place where we safeguard something, but a lot of other things is happening in the forest, and we have a lot of resources that we just haven't in our hand as a gene conservation unit. Well, now actually many years later, we included 82 species, and most of them are shrubs. And you can see that we have 20 coniferous trees, not only one of them or two of them are native. So that is a very huge number of species, but we have made 89 gene conservation units, and they are all, every unit have more species in it. They are implemented as part of the operative plans for the state forests, which are revised regularly, which also is important when you try to make such a long-term program. And then well, conservation by use is not a part of the strategy and implementation. But since I'm working in a state forestry also with seedling supply and seeds and breeding, I would say that the concern about the genetic diversity is more widespread than just part of the gene conservation program. Here's a map of where we can find the GCUs, and as you can see now, it's only 58 species. The rest of them are part of the ex situ protections. And it differs from species to species how well they're represented. Typical for forest trees, they are represented at between 8 and 15 sites. And very often the GCUs are covered of other kinds of protection because we found that was a way to secure the long-term protection of the areas, that it perhaps also was part of a natural conservation of a nature type which were represented by some of the trees that we also wanted to protect. In our management guidelines, we emphasize the natural regeneration, and we have put on an isolation zone so that if we have neighboring areas within the state forests where we do some plantings, we need to use the local material for this planting operations. And then this about the long term, well, in situ protection, dynamic in situ protection, of course, could be going on forever, but we must also be realistic to say, well, this continues until other decisions are made, and hopefully it will be possible to continue with the ownership and with the plants and with the way the areas are protected, but the future, of course, will show. Then when it comes to ex situ, we have seed stands and seed orchards and so on, and we have clonal archives as part of the breeding programs, and that is what we consider as our ex situ conservation. We have 53 species that are covered by that. And well, the clonal archives, at least, they are static conservation and they are temporary. That is typically for a generation or things like that. And also the seed sources in production, well, they also are a temporarily conservation. And in both cases, we must make decisions about regeneration or renewal from time to other, and at least at the end of an ordinary rotation period. And you could somehow here also have this perspective that actually this ex situ seed sources, they really contribute to conservation by use, because the seed harvest will be part of the future forest in Denmark. And you can say that about the value of gene conservation, well, the seed sources, they covers what we today find very valuable, at least from a production or afforestation or reforestation perspective. Yeah. Then I would tell that we find the European cooperation very important. We have been part of the UFGIS and UFAGIN cooperation as long as it has been going on. Actually, our conservation program started before we made this European conservation cooperation. And as you perhaps all are familiar with, there are some UFGIS minimum requirements, and it's mostly about area size and number of individuals and if they can be regenerating, but they are not so, there is not any characterization at the genetic level. But just right now, and actually later this month, the second phase of the UFGIS cooperation will make a presentation of the new status. And one of the things is that there's more characterization of gene conservation units. And if I understood the new web page right, then there is about 350 GCUs that have some kind of characterization. And that is less than 10%. I think we have almost 4,000 European gene conservation units. But I took a screenshot of a map of ABS alba, where you can see that making research or characterization across country, then you can find this is something about the admixture index and proportion of ancestries. And then you can see for ABS alba that there are some groups where the gene resources in different countries differs, and somewhere you have a mixture and somewhere you have more special gene pools. And this is very important for a national perspective to know, is the Danish gene conservation actually very important from a European perspective, or is our neighboring country actually having things in abundance, so we don't need to worry that much about conservation if we only have a few stands or things like that? Or are we actually having something very, very special that we should be very eager to protect? Because gene conservation is also a question about resources, and the implementation in Denmark also reflects that we shouldn't use too many resources on the protection, but we should try to do it simple and do it wise. So there is a need for this European cooperation and knowledge sharing, and also this overview that we can share with each other, so each country can be part of the common conservation, but also be responsible for what is really important nationally and at the European level. Then I have at the end some question about how good is the protection. And this question Erik also raises as this natural evolution, well, will it actually be able to occur in our cultural forms of forest, or will we feel that we have to manage? We must do something if there's signs of bottlenecks, if the ash is dying, what should we do? And also the question about gene flow, which Erik also mentions. Well, actually, somehow we are preventing diversity by trying to make isolation zones. And perhaps when it comes to the wind pollinators, the isolation zones aren't that effective. But knowing if it's a problem or if it's important. And then, of course, this is about disasters, forest fires, climate change, or ash died back. How should we react? Our protection can do something, but it can't do everything. The quality of the indicators I've already mentioned. Well, we can count the numbers of trees. That is okay, but that's perhaps not the whole thing. We would like to be able to make informed decision making, and perhaps now 30 years later we will be able to make even a better strategy, or at least with more knowledge. And some of the things that were discussed 30 years ago are still relevant. This is about how much management should we do, how much about should we let the evolution be running, and also the native species, what are they adapted to? Will they be adapted to the future? If we in Denmark say that our neighboring country would need some of the gene conserved, then perhaps if climate change very fast and we lose some of the genes conserved today, then perhaps our neighbor would actually be able to use, or would be glad that if they were concerned, and we just not said, oh, they can't grow in Denmark any longer, it doesn't matter. yes, and hopefully diverse gene watch can improve our knowledge of some of these issues. I look forward to the discussion. Thank you very much.
Thank you very much, Gunnar, for a nice presentation and very informative. I think we have time for 1 or 2 short questions, if there are any. Yes, Darius?
Yes, just maybe very short question. Not to do you have these problems that we have with overlapping of gene conservation units with nature protection areas, which are very strict, and in a way we can't do any management to preserve progeny, you know, to continue this dynamic conservation approach. So how is it in Denmark?
Yeah, actually, we are able to do management if they are still fulfilling the idea about the protection. So we can harvest seeds, or we can do things like that. The protection isn't that prohibitive until now.
Any other questions? I cannot see any. Another one from Darius?
Yes, hello, it's another Darius, but yeah. Thank you, Gunnar, for nice presentation. Just also one short question regarding the GCU selection. Is it still you are using the same approach that you are establishing GCUs in state-owned forests, or it's still or it's already differently? And how many you have private and state-owned? It's interesting also. Thank you.
Yes, actually, we haven't made new GCUs for 20 years of things like that. When it was implemented, then that is what we have been working on. So we have no GCUs in private areas, but for a few species, we know that there are a nature conservation where we have species of our interest. So we kind of say, well, then we don't do any efforts to try to protect that, although we haven't a state-owned GCU, but they have no formal status in our program. And the other question was about the state forestry. You could see the map. There were some spots where there are no gene conservation. That's because there are no state forests at that particular sites. And the state forestry is about 15% or 17% of the forest area in Denmark.
Rosario has a question as well.
Yeah. Thank you very much for a nice informative presentation. I have a question about the EUFGs. I'm not familiar with that. Is it Sweden also part of it? Because in the map, yeah, it is. Okay, yeah. But is it something that if one identifies a unit that should be included, it's just about telling, or how is the process to declare a unit as part of EUFGs, a new one?
Yes, there's a national focal point, so it can differ from country to country how to have new things in, but there's a national coordinator that you can find on the webpage.
Okay, yeah, thank you.
Great. I think maybe we can move on. Yeah. And then if there are any other questions, you're welcome to write them in the chat. Yeah, and I would like to welcome again Rosario Garcia Gil from the Faculty of Forest Sciences in SLU, Sweden, who will present now. Thank you, Rosario.
Yeah, thank you very much for the introduction. Yes, I'm Rosario Garcia Gil, Associate Professor at the Faculty of Forest Sciences in SLU, and I will focus on Scots pine and Norway spruce, which is really the major conifer species mainly for production, for economic aspects, but also because it's so abundant that, of course, it's part of the ecosystem and it sustains a lot of many other species, so it's very relevant. Now I'm trying to move this. Doesn't want. Okay, now. So the overall goal of genetic monitoring, of course, in Sweden and beyond, is to achieve a sustainable forestry. So conservation of genetic diversity is the first thing that comes to our mind, we geneticists, most of us. So it's nothing new. But what maybe is a little bit new is how, for some of you, is how forest in Sweden looks like. Because when it comes to conservation, one thinks in terms of a lot of set-asides and untouched natural forest, but that doesn't exist very much in Sweden. There's a little bit of natural Scots pine forest in the border with Norway, but beyond that, natural, I mean, in a very strict way, you don't find much. We have some set-aside forests, but I'm not going to focus on that because actually there is monitoring there, but it's not so much research on the level of genetic diversity in those set-aside biotomes areas. But this is how it looks like, the forest in Sweden. So mostly owned by the public, which might be different in other countries. Most of that forest is managed, about 62%, and mainly by, about 23 million hectares. And most of it is managed by rotation forestry. And that means breeding, so selecting the best, the plus trees, deploying them in seed orchards and clonal archives where we have our plus trees that are crossed constantly to be tested. So a lot of rotation forestries are clear cuts and regeneration with improved material, actually from 80% to 95%, I would say, depending on the species. The regeneration is based on seeds or seedlings from seed orchards for improved material. So the factor of the human factor, the anthropogenic factor, the breeding factor is something that is a concern in Sweden. Of course, if you think in terms of pest attack, that's the main concern. But I'm not a pathologist. I only collaborate with pathologists, so I'm not going to focus on that question. So if it comes a pest that erases the entire Norway spruce or Scots pine, there's not much to do. But let's imagine that will not happen. So let's worry a little bit what is more realistic, which is breeding. And of course, Eric gave an introduction that helps my presentation because I also think that we should think in terms of neutral genetic diversity versus adaptive genetic diversity. And if we look at pine and spruce genetic diversity, even at the Euro-Asiatic level, the numbers, and there's many publications, mine and from others, it shows that there's plenty of genetic diversity. So that's not, I would not consider very much endangered. The effective population size is large. There's a lot of this long distance crossings and a lot of recombination going on. So that's not the issue here, I think we should worry about. Then, despite of so much genetic diversity, the populations are very similar. That's the figure in the right. So very little diversity of the variance, as you can see from the principal component analysis, is 0.7%, the x-axis, and 0.47 in the y-axis. So basically, it's like one panmictic population, and then we keep seeing that. But then let's look at the adaptive genetic variation. There is where we see differences between populations, like dramatic. I mean, especially if you look at the latitudinal variation, well, at the neutral level, the trees look the same. Then actually there is something behind, some genes or group of genes that might differ because we see perfect differentiation and clinal variation for adaptation to frost or light, being day length or light quality, which has been my topic since 2000. And actually sometimes we can see the genomic signal. For example, here in the phytochromes we saw from north to south of Sweden, we see a perfect variation in allelic frequency. So certain for that gene, certain allele increases from south to north and the alternative one decreases. And we see that for many genes, actually. We are keep working on that. And that is the adaptive genetic variation is the one that I worry most for, actually. So when it comes to conservation efforts, I think that one of the aspects to investigate is if the impact of breeding on the effect on genetic diversity, because we want to preserve the capacity of our forests to respond now and in the future. So we need to keep that genetic diversity in good shape. So, for example, we conducted a monitoring genetic assessment of natural populations. Wait a moment, because this is in the way I don't see even the graphic myself now. So we have natural population, and these codes find are really natural populations close to Norway. We have bred populations. So they planted as a result or replanted from improved material after one cycle of breeding. And even we sample also ancient samples, and that means 500-year-old or more. And we sampled from middle-aged churches and houses, and we could extract DNA, which was amazing. And if you look at the figure in the right, what you see is that the red dots are the ancient populations. Like you could imagine that, or it represents the genetic diversity 500 years ago and beyond. And they sort of group apart, you can see that. But when it comes to the triangles and squares in the right, that's the bred and natural population. What we see is that there is more genetic diversity than it was before, if this is a good representation of what is existing now. And not only that, is that there's not a big difference, at least not at the neutral level. So this is just microsatellites. But we were quite impressed that something we have done that has increased the genetic diversity. Of course, we know we have been bringing in material from other countries, so probably that's one of the contributions. But now, that was the neutral genetic diversity, and I also mainly concerned about the adaptive genetic diversity. And for that, in the same populations, the natural and bred populations, we compare their level of response to drought. So we collected cones, extracted seeds, produced the seedlings, and established a common garden experiment in the greenhouse. We even genotype also those trees from the natural and bred populations, and we didn't see any much strong differentiation, very small, like the usual. But what happened with the response to drought? Well, this is just a representation of the common garden experiment when we had images from RBG cameras, time-lapse data, and also thermal camera images. We assessed the canopy, but also the roots. And then we created a drought response index. And to our surprise, after one cycle of breeding mainly for volume, for growth, what we see is that the blue dots, which are the bred populations, actually responded somewhat better to drought. They have a little bit higher tolerance to drought. So under drought condition, they grew the same or a little bit slightly better. That was surprising. However, that has a price. So the selection for volume seems to have been changing the fibers or the structure or the number of fibers that has been increasing the response to drought. However, at the cost of decreasing the level of genetic diversity for drought response. So this is the price we are paying. So breeding, of course, is affecting volume and something more than volume. But then we are also investing quite a lot in developing tools, methods and tools to assess genetic diversity for adaptive side. And for that, well, we are, like many of my colleagues here in the audience, I've seen your names, are utilizing remote sensing tools, like, for example, the LiDAR, but also the multispectral, and we are also now testing the hyperspectral. And it's not only to assess form and volume and quality, but also, for example, to assess timing of bud flush, which we are trying to connect with resistance or tolerance to certain fungi. Because if the trees are flushing the bud exactly when, or synchronized with the moment when the fungi is producing the spores, then they are more likely to be infected. So, for example, so this is a biotic stress, but we are also trying to predict wood properties just with RBG camera images of the bark, because wood properties are heavily related with drought tolerance, for example, but even with cold tolerance. And with a small data set, which we are now increasing, we could see that actually with RBG images of the bark, we could somewhat predict velocity. It's not very high correlation, but there is hope, let's say. We are also now trying to develop, actually, we also trying to develop methods for early selection for improved material for drought. So we have been correlating data for drought index in the greenhouse and in the collecting also increment core from the mother, from where the progenies come from, and try to correlate the ranking for drought tolerance based on ring analysis on the mothers and the ranking in the greenhouse of their progenies. And what we can see, and this is very preliminary data, it's very fresh data, is that there's certain level of correlation between the ranking of the mothers for drought tolerance if we try to predict them. So we can, there's some level of predictions we can do from assessment at the nursery level on how the trees will look like in the future for drought tolerance. And we're also trying to utilize a tool which I think is really, really useful, is the resistograph, which usually is for the measurement of density across the annual rings. But there's already some work that indicate that it's possible to translate this data into annual ring width, which again can be translated into drought indexes. So we are trying now to develop a model for assessing the trees rapidly in the forest for drought tolerance using the resistograph. And of course, applying this it can be applied to natural populations, of course, but when we apply it to populations where we have a progeny structure, like in progeny trials, which is very abundant in Sweden, then we can also make estimations of the heritability and rank or identify superior trees. And I think that was, I don't know if it was too short or I was too fast, but this is a little bit I wanted to acknowledge many of the colleagues involved in the efforts being done to monitor genetic diversity, especially for the adaptive side in Sweden. Thank you very much.
Thank you very much, Rosario. Very, very interesting work and findings. We already have questions. Yes, Darius.
Yes, thanks to Rosario, to a very interesting presentation.
Hello, Darius.
Hello, yes, so many interesting data. Just a short response would be a question. Do you think this all-neutral variation, isn't it correlated very much as regards diversity with the adaptive variation? So that if we score it with neutral markers and get the variation higher, that also works for adaptive variation. What is the connection? What do you think? How tight is the connection?
Yeah, well, that was discussed actually not so long time ago with in a conference in South Korea with Santiago Gonzalez, and I mean, population geneticists would usually rely mostly on neutral assessments of genetic diversity or assessment of neutral genetic diversity, and the conclusion is that it's not that direct connection always. So trying to make predictions with just neutral genetic diversity, it may work, but it's not straightforward, and there's many times when the picture that you obtain is not correct.
Any other questions? If not, we could move on with, yeah, I don't see any hands, with Barbara Fusi from the Bavarian Office for Forest Genetics from Germany. Barbara, whenever you're ready.
Trying to share. Hello, everybody, trying to share my screen.
Yes.
Can you see it?
Yes. Yes. Looks great.
Then let me start. Thank you for the invitation to talk about our experiences with forest genetic monitoring in Bavaria. We have started about 15 to 20 years ago to think about genetic monitoring, and from that I can give you some some details. First, let me start with the Bavarian conservation concept for forest genetic resources. Here you can see in the graph, it consists of 9 steps that if we think about conserve stands or populations for a certain tree species, which we follow. So first we start with the species selection, distribution, and stand assessments. Then for all the populations, we do, or for the potential populations, we do genetic sampling, but also like observations of phenotypes and vitality of the stand. The representative sampling within the plots usually consists of 40 to 50 trees. Then we do genetic analysis and comparing diversity values among populations, but also compare with existing seed orchards or newly selected plus trees. Also from the genetic data, we delineate gene conservation regions. So if there are differences between the gene pools, we also consider this when we propose the conservation regions. And this is followed by priority setting and for worthiness and urgency of conservation measures for the certain species. And then we select conservation measures in situ or ex situ or both, depending on the species. Yeah, and so I can give you, sorry, one example. This is Ulmus levis, a rare tree species not regulated under forest reproductive material law. So when starting a study on a species like that, there is a lot of missing knowledge about use and conservation, but also of occurrence, distribution, population size. So the first step is to gather knowledge on the distribution of the species. As I said, we take samples from all the potential conservation stands. And here you can see the results. We usually take about 15 to 20 populations per species. Here you can see that there are clusters in the north following the main river systems, because Ulmus levis is a species near the rivers. And here you can see in the north and in the south, there are different gene pools. And so also we followed this structure when we delineated provenance regions and gene conservation regions. And here at the lower graph, you can see the table, you can see here the values for genetic diversity and allelic richness. And for example, when there is low values, we do not propose them as seed stand or gene conservation stand. As part of our conservation strategies, we see the genetic monitoring, which is for us the assessment of a forest population's capacity to survive and reproduce and persist under rapid environmental changes on a long-term scale. This definition has been developed in the Life GenMon project, which was a EU project where we took part together with Slovenia and Greece. And most importantly is the periodic assessments. So certain measurements, for example, observations or samplings, have to be done regularly depending on the parameter that needs to be assessed. It can be every year, 5 years or 10 years, but a regular scheme is needed. So the actual ongoing genetic monitoring at AWG started in 2006, when a first observation plot for Fagus sylvatica was established. You can see a picture of it here. 10 years later, the German project GenMon was initiated with 14 and 10 plots for beech and spruce covering, also in Germany, whole Germany, covering managed and unmanaged stands for beech and spruce. And in Bavaria at AWG, we have 2 plots in lowland and mountains. We observe since 2006 and 2016, 1 Picea abies plot in the Alps, and the Live GenMon project I already mentioned before. In this project, we initiated 1 plot for Abies alba, and we are observing it since 2014. Here you can see the distribution of the plots of the GenMon network. You can see it is regularly distributed over Germany. And here we have observed bud burst for 7 years now, and also did genetic analysis. Here you can see as an example, one of our plots for Fagus sylvatica near Munich. You can see the plot design. So there are around 250 adult trees spaced over about 4 hectares. In the 1 hectare zone, all the adult trees are marked and sampled, and also natural regeneration plots have been installed to be observed and sampled. Then we have seed trees, 20 seed trees where we harvested seed, as I said, in 2006 and 2016. For these seed trees, we also observed the bud burst on this. And now I will come to some results that we gained from microsatellite analysis to compare the genetic diversity. For example, you can see here in the graph a comparison between seed lots of 2006 and 2016. This was because we, as I said before, we wanted to observe the population's capacity to survive and reproduce. So for the reproduction, it's important how the composition of the seeds is. And then when you compare these two, you cannot see really a lot difference in these 2 years. And the values here also do not show a lot of difference. That's why you can see here on the right side 2 graphs for the dominant pollinators in the 2 years. Here you can see the number of pollinated seeds, and here the trees that were the pollinators. And when we compare these numbers, we found that these are quite the same in the 2 years, the total number of pollinators, but also the number of known pollinators. So the number is almost the same in both. And then you can see here that the same pollinators in 2006 and 2016 are 51. So there are some pollinators, probably the most dominant ones, which are the same in the 2 years. Please keep in mind these results, because I'm now showing you a second example in Abies alba. but also I chose this example because this is independent from the genetic markers. So the if you have the parents identified, then it's you're not it's not important which markers you use for this. This also is important if you do long-term comparison, because if the number of markers change or the markers itself, the genetic values cannot be compared anymore. So that's why in this case we use the pollinators or the parents. On the ABS alba plot, we have adult trees and seedlings from different years, from 2015 and 2019. Also, you can see the absolute numbers here are more or less the same. In this case, only 13 trees were the same in the 2 years. So one could ask, is it good that there is some change, because then there is a different genetic diversity in the offspring, or is it a sign that we should think of because there is a change? So maybe this can be discussed later. Here I want to show you results from a project called PhenoClim, where we analyzed data that we got from Genmon plots, as I showed you, distributed over Germany for 7 years of observation. And you can see here on the right side, the first graph is for beech. You can see that the bud burst for many trees is the same over the years. You can see here the green one as one of the first flashers. So they keep more or less the pattern keeps the same. So also pointing to a considerable genetic basis. The period when the flashing happens varies from year to year. As you can see here, there is only a short period from beginning and ending. In 2019, it's quite a long period from the first flashing tree to the last one. Also here for spruce, the patterns, you can see that this is not so variable compared to the beech plot. In PhenoClim, we also looked at the temperature reactions with regard to bud burst. And here you can see with increasing temperature across the plot, beech reacts with starting 4 days earlier per degree of higher temperature. So you can see that when the mean temperature goes up, that the beech flushes 4 days earlier. For spruce, it's a bit less. It's around 3 days per degree of warming. And you can see also, because the brown dots are for the higher elevations, the temperature sensitivity increases with elevation, and towards the east in Germany also, it's the opposite, the temperature sensitivity decreases, and this means that in the higher elevations the population needs to respond more intensively. So with this I already come to the conclusions. For the genetic monitoring, it's very important to have long-term concepts. but also with different levels of intensity. But it has to be performed continuously, because if there are a lack of data, because there is no finances or human resources, it's better to do some basic assessments, but this needs to be performed continuously. So as I mentioned already, the finances and human resources have to be secured for a long term. Otherwise, it will not be feasible. And long-term data, of course, needs to be acquired, analyzed, and published. Otherwise, also well, it's not, you cannot argue for a monitoring if there are no results published. So test the concepts and parameters and re-evaluate, because if after a certain time there it cannot be seen any meaningful results, it should be reconsidered. But also needs to be refined the traits and markers for the observations, but also to look for new markers which could be combined with satellite data. This could also give some better insights, or for the satellite data, there is a much longer time period where for example, the bud flush could be observed instead of the 7 years that I showed you for the bud flush observations done by people going out in the forest every spring for 8 to 10 days. So short outlook, what we also did is we established genetic monitoring plots in Ukraine and Iran with adult trees and natural regeneration. This is, I think, important to compare the variation in the managed and unmanaged stands. Here, for example, it's a biosphere reserve, the Uholka, which has been conserved for a very long time. And yeah, here are some of the results for the basic assessments. And with that, I want to thank you for your attention.
Thank you very much, Barbara, for the very interesting presentation. I think, yeah, we can take one very short question. There is.
Yeah, sorry, it was a very interesting also presentation. Many things I would like to ask, but shortly, what you would be thinking about repeatability on the same adult trees? Sort of you come, do you do genetic monitoring in 10-year interval, you come to your site, and have you been thinking about adult generation? Because these will be the same adult trees. So maybe we don't really need to focus on the same individuals. How is with that on your your thoughts on that approach?
Yeah, not the same. You mean, well, we collected seeds from the same individuals. It's not to analyze again the same individuals.
No, seeds are okay, no? Because it's the progeny and that may change. Yeah. But the adult generation that you would do your monitoring in 10 years, do you do on your adult generation?
No, we don't. We do not do genetic analysis again on the adult trees. Sorry, maybe this I didn't mention very clearly. We sampled the seeds from the same mother trees and used paternity analysis in the 2 cases.
Right. Thank you again, Barbara. And with that, now we'll stop the recording, and I will introduce the project, who will give an introduction to the project and its aims. Thank you, Darius.
Okay, so thank you. Do you hear me? My voice is a bit bad today because we have
We hear
lots
you
of
pretty well.
students running around, and as we say, we are suffering from all this, diversity that we have to. Yeah, but hope you understand me. And then I put on slideshow mode. Yes, I think I have shared my screen already. And I press slideshow, and if it is, can you see it now?
The presentation mode.
Yes. You see it in presentation mode. No, okay. Okay, so then we stop sharing. So then we maybe share the yes, just a second. We share the screen. And
you have to go to the three dots in the
yes,
so it is in the same stage you were before. You have to go to the three dots.
But before that, I think I have to share it. So
Yeah, you can. Did you share your a window before or the screen?
yes, I just wait a second. This is bad day today. All these electronic things, so I share it again. Share. I think I okay. And then I slide show. And three dots.
Yeah, then you have to hide presenter view. I think that will work. No, that doesn't really work.
No. Try again.
That's where the slide mode spot. Slideshow
go
starts
to-
with paint.
Yeah, okay, we have two screens, so that's the problem.
Yes, yeah.
So Darius, my suggestion would be that when you start
Let's try
sharing,
one more.
Otherwise, we can follow anyways. I mean, it's up to you.
Okay, so
There's no problem.
I just enlarge maybe this one and
Yes.
Yeah, that's perfect.
Okay, so I will talk about the Virgin Watch, our project. Try to be in 20 minutes, so even earlier, so that we have a discussion on important issues for us. What is this workshop meant, and also thanks to Eric and his team, to Jill, organizing this. Just few issues before. What we face, as we talked about, gene conservation is now, as you see, example of good genetic monitoring site where you have two generations or even more generations overlapping. So we think there would be need for research and cooperation on how to really apply this close to nature management to our gene conservation units to have these age structures formed in sort of very natural way. That we were thinking to show to other colleagues and to call for maybe common studies or common observations on that, on how we really do that. But coming to our project, the idea for that was that we already understand our diversity quite well, and you presented many studies early, and the present studies, of course, even improving this former knowledge. We have conservation strategies for diversity also well developed, or for gene program and so on. And also our breeding is improving adaptedness and so on. But what we feel is really lacking is these early warning tools, or sort of sensing how well our system works. And Rosario, of course, presented interesting data when they compare diversity between improved and natural material, but also this idea of diverse gene watch also to try to work together around the Baltic Sea to establish this early warning system for genetic diversity loss, and then use it together to improve genetic diversity. And of course, just a slide to show that science moves on, and in many countries, this gene conservation is given the duty of some sort of agencies under the ministries. And sometimes they really see that science already far went into the future, and they would be needing and would be interesting to apply new tools in their gene conservation programs, such as remote sensing or DNA marker sensing. So that was another push to develop that system. Also, we had this Live Gemon project that showed they already doing something in the south, that we most probably should initiate something also in the north. And then I jump into our project directly. So basically, it is called the Diverse Gene Watch. It has a longer name. It started in 25, and we have 3 years for creating this system. The project is focusing on three species, if you want to have the precise aim, so we want, as Interreg describes it, we would want to team up scientists, also stakeholders. It's very important to create, pilot, and implement durable transnational genetic monitoring system around the Baltic Sea. And basically, we would like the system to effectively estimate genetic biodiversity, I call in this way, and timely warn on coming declines of genetic diversity in the keystone forestry species. And we have 4 species for that. We got money in that project, is Tilia cordata. Of course, we could comment later, well, why we chose those species, but you know well all those species. So Quercus robur, Pitia abies, and Fraxinus excelsior. Fraxinus excelsior is very interesting, what diversity remains after all this dieback. And for instance, we now, when we looked for our genetic monitoring sites, started to see new regeneration coming, and quite in larger quantities. So maybe it's recovering, and it's very then interesting also from scientific point of view to have assessment what happened, how narrow is the bottleneck, and so on. And here is the general scope of the project. We have institutions from Sweden, that's Lidenius University, then we have Germany, then we have Poland, and 3 Baltic states participating in that project. Only from Germany, we do not really have a scientific institution, so that we maybe, as Barbara and Birte is there, maybe they would be interesting to do some follow-up and so on, or do something similar as we are doing. And okay, so what we will do, basically in the project we have 3 working packages that are sort of predefined by Interreg. One working VP1 is basically we create all the theoretical background for genetic monitoring. And basically, we focus on the system by itself, how we will do that. And then we discussed on DNA marker systems we will use. And also, we would like to test cheap alternatives to DNA markers, and this is remote sensing, in aiming to estimate genetic diversity, namely genetic diversity. We don't know would it be possible or not, it's just sort of try. Then also phenotyping, and basically it's phenology, and how to translate all this phenology, like also Barbara did, but how to translate it into genetic diversity estimate, to have a statistics for that, and then compare that with DNA markers and see on whether we can use it as a cheap alternative to do more frequent monitoring if there is a need. And also not so simple how to enrich genetic diversity if we have problems. There's also some technical issues. So all this VP1 is a theoretical part, and half of it it's already finished. We have this as we call deliverable D11, which I will present shortly. Major things are presented there, several alternatives. We have to do some piloting here, some sort of small scale studies in order to decide on the final guidelines, which we'll have to finish by February next year. And then in VP2, when we have all this, so to say, monitoring guidelines, then we'll do some monitoring, piloting of monitoring in each country. We will do, we will select 2 monitoring plots, and then I will show later what we will do in either of them, but we will do basically, we will do DNA genotyping, Then we do remote sensing in one species, and then we also do phenotyping in all plots. And then we compare these results, adjust the technical guidelines, and then in VP3 sort of we issue the final final solution for the Baltic Sea region. Then we have the thorough plans for each activity. Basically, then I go to the guidelines, what we have already made and how we are planning to do that. So short summary, I will not be in time to finish all, is that we select genetic monitoring sites and genetic monitoring plots. And in each plot, basically, it will be a defined area, maybe 5 hectares or so. We discuss the size depending on the species. And then we select 2 generations, adult generation and progeny generation. And then we do DNA genotyping of each generation. And then for one species, I think it's Norway spruce, we will do remote sensing of the same trees or the same site. And do the phenotyping of the same trees on the same site, and we do that in simultaneously, sort of in each Baltic Baltic country. And we try also an effort to connect it with biodiversity schemes. That means genetic diversity is connected to how we will to sort of monitoring of biodiversity, like Biodiversa Plus and so on. Okay, so just conceptually speaking, it's sort of very important also for us to define what we are looking for when we do this genetic monitoring. And as Eric also put these questions for discussion, what I was thinking is that our goal maybe is sensing the genetic evolutionary processes within the population. So, you know, basically effects of selection, drift, and so on, and timely alert on approaching genetic diversity loss. Sort of we concentrate on those tasks. And then third, to apply genetic enrichment measures. And so going into more narrow details, we will follow evolutionary processes by monitoring, as it could be viewed, mating patterns among the parents to be reflected in the progeny. And is something wrong with the mating of the parents that we get a reflection in the progeny. So we sort of try to sense these deviations from random mating due to genetic diversity loss, and neutral markers, of course, very good for us in that sense. We define progeny class as of 2 to 10 years old, so that we consider multiple sort of mating events, not only one mating during 1 year, but when we have this range of 10 years, we sort of level out it and consider multiple mating events over the decade. And then for the parents, we choose the overstory mature age class within the years of 20, sort of also that they should really avoid in this sampling generation, they should avoid parent progeny, you know, presentation in one age class. So this 20, but not more, what I wanted to say. It's hard to maybe to assess that in adult trees, but it's some technical issue. And so, okay, we select those plots, we select those 2 age classes, and then for each we do genotyping, as I call spectrotyping, and morphotyping or phenotyping, and then compare the results, and based on that develop the genetic parameters and make solutions on what is the genetic diversity level. I don't know, just check the time, how much. I think I should finish at 12, so.
No, 4 minutes.
4 minutes. Okay, but I think I started later. I wrote, okay, I should finish 11:40. Okay, so sorry. Okay, so basically this, then I skip this slide. So we're going to do what we decided to do, how our network looks like. So each country establishes those forest genetic monitoring plots marked here by FGM, and then you have the advisory board in each country, just a board of scientists that could advise this institution which accepts to do genetic monitoring. Yes, and then once per 10 years they do this genetic monitoring activity simultaneously, issue report and send it to Central for a genetic monitoring institution, which also has sort of central advisory board. This institution summarizes the report, presents it in a conference, publishes it somehow, and basically this international board of scientists make a decision: what is genetic diversity loss risk? Is it low, medium, or high? And then based on those algorithms, low, medium, or high, we make an action plan on genetic diversity enrichment, how we do this for that species. And that's very important message we wanted to say to all this, that in order to work in long term, we would like it to be this genetic monitoring to be officially accepted as a part of gene conservation program. So that if you have gene conservation program, we have dynamic, static, as you presented here, but also in the parallel, we have this genetic monitoring unit, which sort of is an integral part of your gene conservation system, which brings innovation, which brings monitoring, follow-up, and sort of is very important then to have it enlisted in that. And then another slide to really show now overall how we do that in the time perspective. So this is our network of phylogenetic monitoring around the Baltic Sea, and then we have monitoring by DNA marker each 10 years. So if we do it in 25, we repeat it in 35. And each 5th year, we suggest to do only remote sensing and phenotyping if we succeed with developing these methodologies. And then we have this, as I explained, for the genotyping location. Then we have genotyping, remote sensing, phenotyping. We have reports. We have conference, and then we have action plan already prepared, depending on which light we give, green, yellow, or red. Okay, so basically that it's ended, and then we have a lot of prepared some methodological details, how to sample, on whether to have it round sampling plots, or you go by zigzagging in 20 meters zigzags and so on. Phenotyping, we also prepared how we should score the phenology for each species. We also remote sensing. Remote sensing will be done only for Norway spruce because it's just a try, just a piloting. And basically with remote sensing, we are interested in answering questions: is it possible, and how large is the error when we have this diversity estimates? And the strategy is to calibrate all the systems that we have on seed orchards, or clonal collections, because then we have known genotypic, constitution. Develop the spectral ranges which are interested for us, and go to over these genetic monitoring plots which we genotyped, and to do this remote sensing photographing from different levels. So basically, I think, and also there are a lot of issues to discuss, as Eric said. Just to stop with that, basically is this 10-year repeatability on the same or different FGM sites. Because, I mean, some adult generation trees may die, and okay, then we resample adult generation and we get a new adult generation in 10 years. But if they not die, then it's the same trees. It's no really point to regenotype them. Then what we all our sensing system will be based on the progeny. But maybe then, just as the population is large and it is sort of, then we could go somewhere and select new. So that's a matter of discussion. Also, when estimating genetic diversity alerts, it's important also avoid genetic diversity outliers. Select really robust parameters that we should really base our decisions on. Also, how to translate phenotyping into genetic diversity estimates. That's a challenge for us. So basically, I stop now.
Thank you very much, Darius, and really appreciate that you were keeping track of time. Are there any questions? Gunnar, you see hand? or Ole, if Gunnar doesn't have a question. Okay, Ole, please.
Thank you, Darius, for your presentation. I'm working a bit with drones myself in a big project we have, but I was thinking, how do you imagine that you can kind of register genetic diversity with these sensors?
Yes, basically it's the connection with phenology. You know, phenology is connected with genetic diversity, and phenology is not only visible but bursts that we, you see in the slide now, that we could capture, but also hyperspectral imaging could actually follow like this. For instance, is a Norway spruce site with different provenances, and already with your eye you can see the differences in their sort of in their color. But we did some studies with Scots pine, and it is possible with hyperspectral imaging sort of to get spectral ranges which separates those populations.
Okay, so
and we connected with phenology rhythm because it continues over all year. All this, you know, internal structures of the cell follow the phenological processes, you know, active growth, lignification, and blah 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 blah. So we hope that we will be able to detect it.
Okay, so you need some traits which are actually have a very high heritability.
Like phenology, and you know, the phenology cycle is very much goes over all the year, you know, dormancy release and initiation of active growth. All this, you know, could be reflected in the needles. That's what we hope. Yeah.
Okay.
Rosario, I think, has a question.
Yeah, thank you very much. Very interesting presentation, Darius. I have several questions, but I will go only for some of them. So when it comes to phenology, we also trying with the multispectral, but you're using the hyperspectral, right? Did I get it?
Yes.
Yeah.
Yes.
Okay, so because you see some advantages, because the level of resolution in principle could be lower because the pixels are larger, to call it something. But so it's a method that you have been testing and it's working. Is that...
Yeah, we have tested that on Scots pine, and it was like, you know, that we detected differences between the populations based on hyperspectral imaging, and that was from the airplane, I think.
Okay.
1 kilometer or so at the height. So that was quite, you know, amazing. That was not drone and an airplane. Yes.
Yeah, I'm pretty amazed. So I guess you are basing that on changes in color and not in the segmentation of the buds, and then later
Yes,
on.
most probably, yes.
Yeah. Then because you're genotyping this many number of trees, and you are also obtaining phenotypic data, while the main focus is to monitor and assess if there is sufficient genetic diversity, is also an objective to select already even superior trees plus trees and establish some seed orchards or...
No, just the focus for us is monitoring, sort of assessing genetic diversity, you know, that then yes, sensing the drop when we compare progeny with the parents. Yeah. That's the main.
Because I guess with the molecular, with the genotypic data, you could build a G matrix and then that you could even rank maybe some of the, if there is some clear causality between them. Yeah.
We could consider that also.
So breeding without breeding, landscape breeding. Yeah, I have more questions, but I think I leave it here. Very interesting.
Thank you. And thank you again, Darius. We are 10 minutes late, but I think it's not that bad. So I wanted to thank you all. And I think now it's maybe time for a break, 15 minutes break to stretch a little bit and get some coffee.
10 minutes. So we, yeah. Okay. We meet 5 past again?
Yes, we can meet 5 past again. The meeting will be on and open.
Yeah.
And yeah, I'll see you in 10 minutes again. Thank you. Everyone, I hope you had time to grab some coffee. We'll move on to the second part of the online event now, and I would like to introduce Jill Olofsson from Copenhagen University, who will give a presentation on the genetic markers for monitoring the previously mentioned species in the context of a Diverse Gene Watch project. Yeah. Jill,
No.
whenever you're ready.
Is it not working now?
It
Yes,
is working.
It is working. Yeah. All right. All right, so thank you very much. I will, my background is in the molecular genetics and the population genomics. So what I will do is basically to present some of the thoughts we have had behind some of our selections or some of our suggestions for selections so far, but also putting it within the scope of this project that we have. Let's see. There we go. So as Darius just showed, this is basically at a glance the Diverse Gene Watch program, where the aims and the purposes are both to try to do the genetic diversity monitoring, but also at the same time try to look at these evolutionary processes that both Eric and Darius have talked about. So the idea is that we go into the conservation units, and this is just a summary from UFOGIS for how many genetic conservation units of the 4 selected species there is within the 6 countries that are contributing genetic data. And the focus of what I'm going to talk about is the DNA markers here. So I'm not going to touch on the morphotyping or the phenotyping, and also not on the remote sensing at all. So again, it's just a bit of an overview of the idea of this forest genetic monitoring program that we have set up. And the Diverse Gene Watch program is basically dealing with this first phase, where the idea is, as Darius presented, that we would go in and type the mature trees in these conservation units, but also the progenies within them. So if we put together what we had or have proposed to do is that we take 2 plots for each species per country that we are sampling in. And then we have these 2 sets of samples, both the adults and the juveniles, and we would take 100 individuals of each of these sample sets. And that means that we have about 400 samples per species per country. So if we now start to sum these up, so we are not going to, two of these species are not going to be sampled in all of the countries, but what we're looking at is that we have about 2,000 to 2,500 samples per species that is going to be genotyped within this piloting part of the project. So that means that we need some kind of system that is efficient, but also cost effective when we do this. So what kind of markers can we then select? And I've chosen to do this in a more historical fashion, where I say that the molecular part of the forest genetic monitoring kind of started in the 1970s, where we could start looking at these allozymes that are really good at estimating the genetic diversity, look at gene flow and inbreeding and so forth. In the 1990s, then all of these PCR-based methods started to come out, where there was an increase in the number of AFLPs, microsatellites or short sequence repeats. And I put single nucleotide polymorphisms in brackets here because you can actually assess them with PCR-based methods, but we're not coming out there in the 90s. So in addition to these genetic diversity, gene flow, inbreeding, relatedness, etc., what these PCR-based methods also allowed us to do was to track changes in allele frequencies and also look at some fine-scale population structures. About 20 years later, these high-throughput sequencing methods or technologies started to become widely available for the larger communities. And there was a switch into looking at these single nucleotide polymorphisms or SNPs in some kind of sense from a high throughput sequencing, whether it's being whole genome sequencing, genotype-based sequencing, these reduced sequencing systems such as RAD-seq, ddRAD, etc., etc. But there are also some targeting sequencing here where you can basically just look at the exomes or the genes within these genomes that you are analyzing. So in addition to all of these more historical or standard measures of gene flow, inbreeding, changes in allele frequency. What these high throughput sequencing methods actually allowed us to do was to look at both natural and adaptive loci at the same time, and then trying to start to associate these genetic trends with climate change, pest and forest management, and so forth. And in a more detailed way, kind of inform these conservation and breeding programs. Well, I think we can all agree that allozymes is a historical marker that we're not going to use. And then if we are looking at this more PCR-based and also the high throughput sequencing, what I've highlighted here is that if we're looking at PCR-based methods, we're mostly talking about microsatellites or these SSRs. Whereas if we look at these more high throughput methods, we're looking at the short nucleotide polymorphisms here. So if we just compare them, what we have with the microsatellites is that they're really good at looking at relatedness, inbreeding, changes in allele frequencies, and we can to some extent track these population structures. And where if we go more high throughput sequencing, looking at SNPs, where we can add to all of these things on the left is that we can have both the neutral and adaptive loci, and we can do these larger scale and genomic analysis looking at the genetic trends. So if we look at some pros and cons with these different markers is that, of course, the microsatellites are highly polymorphic and codominant. They're neutral, so we can kind of avoid looking at selected loci, for example. And they also have, if we have set up panels already or multiplexes, they are have low cost for small scale projects, and they can be developed in-house. However, we know that we can only look at the limited number of loci, and if we need to do some development, there can actually be a substantial cost for these development of these panels. And we also look into these scoring inconsistencies because if we're looking at PCR-based methods, we know that we might have between lab differences in how these different fragments are running on the different fragment analyzers. While that can be solved, it also requires quite a lot of lab work and also manual work for scoring these alleles or these markers. And on the contrary, the neutral markers, while it allows for these non-selected markers, also doesn't really allow us to look at these more adaptive loci to looking at adaptive trends in our data. However, there is, of course, both positives and negatives. We're moving to more these high throughput sequences. On the plus side, we get a higher genomic coverage. We know that they are quite reproducible, and they're quite cost effective per loci. So per SNP marker we type or we get genotypes from, it's actually quite low cost. And we know that we are more in the forward thinking, so we are touching more about what the research community is doing. On the other hand, if we're looking at SNP markers, because they're mostly biallelic, there is low information per loci, so we need to have many loci at the same time. We might require quite high bioinformatic capacity, so it might require more computer work afterwards. And the total cost, so while it's cost effective per locus, it might actually be quite expensive as a whole. So the total cost of these this project can be quite high. And in some cases, we also need higher quality DNA as opposed to these PCR-based methods. And I put in bracket that genomic resources are needed. So for some of these more high throughput panels or solutions, we would need to know like the reference genome. However, that can be overcome in using different methodologies here. All right. So I borrowed from Eric a little bit of this need to know. So if we're looking from a genetic monitoring point of view, I mean, we would need to know the relatedness, inbreeding. We would like to know the changes in allele frequencies. For the type of markers, we want them to be polymorphic and neutral, and we want low cost. So we always want to pay as little as we can. But we would also like to have some be able to inform conservation and breeding programs here. On the other hand, there is also a lot of nice to know things here. So we would like to be able to look at the population structure. We would like to be able to have both neutral and adaptive markers such as we can track both changes, and we would like to be able to test associate these genetic trends with some climate change, pest and management. And we would like to have high genomic coverage, and we would like to be forward thinking or at the forefront of genomic resources. So if we kind of combine these need to know and nice to know, what we're kind of ending is in a little bit in between. So on the one hand, it's nice to have these PCR-based methods. On the other hand, it would be nice to be this have whole genome sequences, and somehow we need to kind of end in the middle here. So if we now look at the main aim of the project and some criteria of this genomic markers or genetic markers. So the main aim is to create, assess, and implement the genetic diversity monitoring system that will estimate genetic diversity effectively, right? And we have some genetic criteria for these markers. We are looking at neutrality, high diversity, ease of typing. But we also have some external criteria here. It should be cost effective, both in total and per locus. And we would like to have quite easy sampling protocols. We would like the analysis to be quite easy to do, because it's fine if we do whole genome sequencing and as a research project sit for multiple years and analyze this data, whereas from a monitoring point of view, we would like to have the analysis out, and as Barbara also said, things needs to be published, because otherwise they cannot be used. And then I've added this add-on research questions. We would like to do these genetic associations analysis as well. So if we combined all of this, what we kind of ended up in our recommendations is that SNP panels kind of, they fulfill all these need to know criteria and provide many of these nice to know opportunities, and it's definitely cheaper and less bioinformatically heavy compared to whole genome sequencing. So if we look at genotyping panels, and specifically at SNP genotyping panels, there is this options to do PCR-based in-house designs. And then on the other hand, we can also outsource our laboratory work to more of these targeted SNP panels. So if we look at some pros with using in-house designs, I mean, the loci flexibility is very high, because we can basically decide what loci we would like to do. Once these in-house panels are designed, they're quite easy and cheap to run. However, we are limited by the number of loci. The developmental cost can be fairly high. It can be very labor intensive, both in the lab and then afterwards in the scoring of these. And we would need some genetic information. So we would need to know which loci we would like to design for. And as I said, we might run into issues with between lab inconsistencies. On the other hand, if we outsource, there might be a reduced in-house labor. We might reduce the between lab differences, because if we outsource to a single company, for example, or a single sequencing centers, everything is processed in that one lab. We might look into these all-inclusive solutions where we would also remove, for example, labor associated with DNA extractions. And we're hoping that this would be reproducible for the future, so that we're hoping that the markers and the panels that we set up today will also be reproducible in 10 years, or if the technology has moved on, if this marker can kind of move with the technologies. On the other hand, we might lose some flexibility in terms of loci selection. That depends a little bit if you're using pre-designs or if you're designing new panels. We are looking at the higher cost per loci, and often we would need genomic resources. So these panels are basically designed based on a reference genome that is out there. So what we have been looking at is some SNP panels that are out there. We have the FlexSeq panels, which are offered by LGC Genomics, and this is the reason why we looked at this, because we got inspired by the work in Germany that Birte has presented here, and there is a pre-designed panel, which we have chosen to call the Degen panel here for the oaks. We're also aware of the Spurgenius project that is running in the southern parts, mostly southern parts of Europe, where they have designed SPET panels with IGA technology in Italy, which is a targeted sequencing panel. So this is this high throughput sequencing with a limited number of loci coming out. And those have been designed in that project both for Norway spruce, for Quercus robo, and for European ash. And then we know that Thermo Fisher has these axiom snip ships that have been designed both for Norway spruce and for the European ash. But currently there are no available snip panels for Tilia cordata out there. So what we have looked into is to compare these kind of already available resources. And if we look at the number of loci, we are the Forgenius, the SPET panels and the Axonum snip ships in general terms offer more loci. However, DNA requirements are also much higher for the example with the snip ships as compared with these FlexSeq panels that we've been looking at. If we're looking at the design flexibility, it basically depends on whether we would like to put efforts and money into designing new panels, or if we are using already the pre-designed panels. So it kind of is quite similar between these three options. The data comparability, I mean, it's also quite similar. It depends on what data we would like to compare with. If we're interested in comparing with the oak data with the German oak data that is published, then I mean, it would also be preferable to use the same markers. But on the other hand, if we're interested in comparing data to the Forgenius project, then using their same panel as they have been using is also an option or also available. If we look at the total costs, I'm not talking about cost per loci here, but the total cost for the whole project, we are actually increasing to the right here. So whereas the FlexSeq is on the lower end, the snip ships are on the higher end of what the total cost for the project would be. And if we're looking at the bioinformatics needs, where the FlexSeq that we have been looking at, the option there is quite low because there is a lower number of markers that are where genotypes will be provided. Whereas on the other hand, the Axonum snip ship provides also genotypes with a large number of markers that needs to be filtered and also processed. And then these SPET panels can be variable because depending on how much you are willing to pay for bioinformatics services, then it can be a low on the lower end of the bioinformatic needs or a very high bioinformatic needs where you need to start with raw sequencing read data, and you need to filter, map to reference genome, call your SNPs yourself, so forth. However, while we would like to choose these panels based on what we really would like to do, like all these nice to know options, what we know is that there are some very strict external constraints here in that we have a set budget. And we have also taken this bioinformatic needs very much into account, because if the idea is that this should be a monitoring process that will be repeated every 10 years, what we would like to have is a fairly easy data analysis that can be used by non-researchers, basically. So we have very much kind of ended up focusing on these more external requirements that we had within the project. And what we have ended up is that we ended on this lower end here, where we are still getting a larger number of markers, but on the lower end of the number of markers, and offering some flexibility in how we would like to choose the markers that we're looking at. Another important reason that we also have looked at is that the companies that we have been in contact with regarding this FlexSeq is that there is possibilities for an all-inclusive solution, which basically will remove the DNA extraction, the laboratory work concerning DNA extraction from our side, which has also been a high priority for us, given the number of samples that we are processing. So what we have proposed so far is that for Norway spruce, we do a new flexi design here. We're targeting about 5,000 snips. The same for the ash. Whereas we, for the oak, would kind of would like to reuse these, what we have called a Degen panel, which has been used in the German forestry inventory, because it's an available panel, and we have data that is relevant for the Baltic Sea region that we would like to compare with. However, for Telia, because Telia is currently lacking a reference genome, it is not possible to do some of these SNP designs. So what we have ended up using is that we will propose to use already designed and already known microsatellite markers. On top of that, within the project, we also have money where we can compare the diversity measures that we get from these new designed SNP panels with microsatellite markers from Norway spruce. So this would also allow us to kind of give a little bit of input to what do we gain by moving to these more high throughput SNP panels compared to the more historically used microsatellite markers. In terms of add-on research questions, with the University of Copenhagen have been very involved in trying to design what we're hoping with these more panels is that we would be able to track some population structures, do some GWAS, because we also know that we collect a large number of phenotypic data within the project. So we would like to see if we can do some genome association analysis and also some landscape genomic analysis, because we can collect the bioclimatic variables from these collection sites. For oak, what this panel is also offering is that we would like be a possibility to estimate the integration from other crocus species within Europe, and also look at the species distribution and hybrid compositions. We know from Bete's work that there is we have the possibilities to do some ecological niche modeling coupled with climate change. And for ash, what we would specifically like to test with these new panel is that we would like to assess the susceptibility of ash dieback, and also to look at the effect of the disease has on the genetic diversity. And for Telia, we are not currently consider a large-scale add-on research question, but we would like to try to do some of these more population genomics or population structure studies using the microsatellite. That should be possible as far as I have. I'm almost done, but I have a notes on sampling here, because we've been talking a lot about what we should sample and what we should not sample and how to sample it. While leaf and needles are kind of the golden standard, depending on if you're a broadleaf or a gymnosperm here, they're usually need to be, or they are best if they're newly flushed leaves, gives the best quality in DNA extraction, but that keeps a very narrow time window for when all of these samples can be collected. What we have good experience with in our lab, and also from talking with collaborators around Europe, is that there is buds give a very good or very high quality and quantity DNA and can be collected over a larger time span. However, we also know that some of these trees are very tall, and while there is methods for either shooting or climbing trees and getting leaves or buds, we do know that we possibly would need to do some DNA extractions from cambiums or from wood material. However, our experience is that we get both lower quality and especially quantity of DNA, and that it's very labor intensive, both in the lab and also in the collection phase here. But what we have agreed on so far is that once we have selected all of the methods that we would, or all of these methods, and the company that we would like to outsource with, is that we will run some piloting on DNA extractions where both leaves / needles, buds and cambium will be tested, and then we will try to then we will give recommendations on what to collect in the field. So basically, just to sum up what I've said is that we think these SNP panels kind of fulfill all these need-to-know criteria, but also provide a lot of these nice-to-know opportunities, and is much cheaper and requires less bioinformatic processing compared to these more high-throughput whole genome sequencing, or even the RAD-seq or exome-seq or anything like that. And what we are really excited about is the possibilities of using these all-inclusive outsourced FlexSeq panels for the free species with a reference genome, because we think that that will be an optimal choice given the number of samples and a large number of labs that are involved in this process. However, microsatellites are still a valuable option, especially for species with few genomic resources here. And that basically concludes my talk. The only thing I have is the points for the panel discussion, and I can show that later if we're moving into that. But yeah, thank you.